Announcing the arrival of Professor Dr. Adiba Kamarul Zaman, the Dean of the Faculty of Medicine, University of Malaysia, who will chair this morning's lecture, and Professor Dr. Chua Kek Heng, Department of Biomedical Science, University of Malaysia. Thank you, and please be seated. Good morning, Professor Dr. Adiba Kamarul Zaman, Dean of the Faculty of Medicine. Distinguished guests, Professor Dr. Chua Kek Heng, Management of the Faculty of Medicine, ladies and gentlemen. Welcome to the inaugural lecture by Professor Dr. Chua Kek Heng entitled Malaria, from Fundamental to Translational Research. Without further delay, I would like to invite Professor Dr. Adiba Kamarul Zaman to chair this lecture and to introduce Professor Dr. Chua Kek Heng.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and good morning everyone. And thank you, Puan Pengacara Majlis, highly qualified Puan Pengacara Majlis, colleagues, dear colleagues. I've always said that my job as Dean has been made easier by having so many talented academic staff within our four walls. I think is the envy of many other medical schools in the country by virtue of us not only having the largest number of academic staff, 564, I think, at last count, but also having the largest number of talented, experienced staff. And without a doubt, Professor Chua Kek Heng, who will be delivering his inaugural lecture this morning, is one of them, one of the gems in the faculty that has been up until he was reluctantly made the head of department and still is very reluctant to helm the department. One of the faculties, if not UM's real quiet achiever. And when I read his biography here, those of you who are not completely familiar with Prof. Chua's contribution to research and to medical science as a whole will agree with me. Prof. Chua is one of our homegrown graduates, having obtained his PhD in biotechnology from in 2003. And his research interest is actually varied, but very, very important, and include studies into polymorphism, disease polymorphisms, mutational investigation studies, and characterization of bacterial virulence factors, and diagnostic kit development. He has attained outstanding achievements in terms of publications, IP rights and patents, and his commitment and relentless hard work has led to more than 130 high-impact publications. His contributions in science and technology are not only limited to novel scientific innovations, but also allow further understanding between human disease and their association with various genetic factors and microorganisms. Prof. Chua's commitment to the study of genotyping of diseases has resulted in the successful mapping of important disease predisposing factors for SLE, for which he published in Nature, is that right? Nature papers, and Crohn's disease in the Malaysian population. The frequencies and distributions of these polymorphisms and mutations in the three main ethnic groups in Malaysia, needless to say, is vital in disease understanding and development prediction, and allow early preventive measures. His innovative approaches, tests, and kits also will allow more cost-effective, rapid, sensitive, and specific detection and confirmation of human diseases and microbial infections. For all his efforts, he has been recognized at the national and international levels, and he currently holds 18 national and 7 international patents, probably one of the highest in the faculty, and 1 trademark and 212 copyrights. He must be a rich man. A diagnostic kit for malaria was developed by Prof. Chua and has been successfully commercialized in 2011. And the kit is the world's first PCR-based malaria detection system, which is able to detect all 5 different species of Plasmodium in a single PCR assay. He has also contributed much to the transfer of scientific knowledge in his capacity as a committee and panel member for the evaluation of numerous internal and external grants and projects, and is actively involved in organizing national and international workshops to introduce up-to-date technologies to young Malaysian researchers and to train young scientists in molecular bench skills. To date, he has delivered more than 100 presentations in various seminars, expos, and conferences, and at the international level has been a reviewer for many international ISI publications. And finally, Prof. Chua has also been selected to represent UM in various major national and international exhibitions and competitions, and has been awarded with more than 90 prestigious scientific recognitions for his perseverance in research and for his excellent research innovations. But what I like most about Prof. Chua's research is that he truly focuses on diseases and developing studies and understanding diseases, and is able to translate it into clinical relevance. And I think, translational research and making your research relevant and impactful is really the way to go in this day and age. And no one exemplifies that more than Prof. Chua. And I think you will hear from him of how his work in malaria, the fundamental research that he has done in malaria, has been able to be translated into something that is now being used in the clinical sense. So ultimately, in research, that is what we're looking for, something that is impactful to society. And I believe Prof. Chua has more than achieved that with his area of work. So, Prof. Chua.
Good morning, Prof. Adiba, and thank you for giving me such a nice introductions. Welcome to my fellow colleagues, lecturers, professors, students, ladies and gentlemen, to my inaugural lecture today. In these inaugural lectures, I would like to share with you how to move on from fundamental research or fundamental study to translational research. And by using one of my studies, research on malaria as the example. Okay, I shall start up with my journey in UM. In 1993, I came to UM to do my BSc program. Okay, so in 1993, I was super fresh. That means I entered the second year for this program. Then I graduated in 1996. After that, I pursued my master and graduated in 1999. And I obtained my PhD in 2003. 1 year prior to my graduation, I was already a lecturer in science fac. That was in 2002. So 2003, I transferred to FON, Faculty of Medicine, due to FON offer me a permanent position here. So I become a senior lecturer in 2006, then AP in 2009, and I obtained my full professorship in 2013. So from my previous study and postgraduate study, I was some sort like interested in molecular study. So those studies are related to gene. Gene is the molecular unit of life. This is actually my own definition. Okay, so a gene is just like is a region of DNA that encodes a functional protein. So at first I thought gene is so simple. It just consists of exon and intron. Exon is the coding region, and intron is the non-coding region. So when you study more, you find out actually a gene is not that simple. Until today, a lot of things haven't discovered, which is referring to the gene. So a lot of things actually need to be studied, from polymorphism to mutation, methylations, or whatever actually are related to the gene before it can transcribe and translate into a functional protein. So I'm some sort like master in microbiology, molecular biology, genetics, and biotechnology. So the field is quite wide. So I use my knowledge in this four field to focus my research on disease and polymorphism study, virulent factor determinations, functional gene determinations. I'm looking at immune cell responses in some of the autoimmune disease. At the same time, I study the gene expressions for certain human chronic diseases. So for the first 6 years of my career in this university, especially in this faculty, I'm focusing on publications. It could be driven by the KPI, as we know. So day and night, actually, I'm working on paper, prepare manuscript, publish article. But later on, in 2008, I suddenly realized actually you have to translate the fundamental knowledge into something applicable or a product to benefit the society. So translational research. This is the outcome of the translational research. Diagnostic kit. Diagnostic kit actually is quite simple. As long as you know where is the target gene, what is the active site for the target gene, you can develop the probe for the diagnostic kit. So similar approach can be applied for vaccine development. So this is another output of the translational research. One of the good example is golden rice. Golden rice actually won the Patents of Humanity Award recently. That means in 2015, and many people are aware that central role of vitamin A is to maintain the integrity of the immune system. But for laymen, actually, we just always think vitamin A is just good for the eyes, especially at night. But the exact role for vitamin A or beta carotene is actually to maintain your immune system. So rice produce beta carotene during the biosynthesis pathway, but along the way during plant development, actually it can't be produced in the grains. So it's some sort like is accumulated in the leaves. So in this group, two genes have been isolated into the inserted into the rice and produce beta carotene in the grains. And based on WHO report, approximately 40% of kids under the age of 5 in the developing and poor country suffer from malnutrition deficiency disease. Therefore, this golden rice is not just an invention, but it really solved the life-threatening micronutrient deficiency problem in the developing and poor countries. The ear mouse. This is the ear mouse actually is a human ear has been grown in a rat by a group of researchers from Japan. The group of researchers consists of people or professors from University of Tokyo and Kyoto University. Based on their research, they managed to grow the ear in the rat. Then they claim that in the next 5 years, they'll go for clinical trial. And this is very important. If successfully, the techniques can represent a new way to help those newborn with anoxia or microtia, where the external ear is absolutely missing or malformed. Okay, now we look at another example is BT corn. We have BT sweet potato as well. BT actually is the gene. That means BT corn, the corn express the BT endotoxin. And the endotoxin actually is try to kill the parasite predators. So it's something like selectively kill the predator, but not really harming the environment and all the mammals, including not only human, including birds and also fish. So this is one of the example of my previous research on tobacco plant. Okay, the initial intention of my research on tobacco plant is not to increase the yield of the tobacco leaves. I should have done that. Then I become the cigar tycoon or whatever. Now I no need to stand here to deliver the inaugural lecture to you anymore. But I just try to turn the tobacco plant into a protein production factory and produce this kind of anti-CMV as CFVs actually is a chimeric recombinant protein or antibody, which is actually useful in life sciences. Okay, a lot of good findings and has been carried out or discovered by all the researchers in this country in general, but the finding is still far away to reach the target. That means is to produce a product or to become an applicable product or applications for the society. Okay. This slide shows you the features of scientific output, and all these keywords that are actually very important if you are intended or interested to involve yourself in the applied or translational research in future. You have to remember all this is something different from what you have done for the fundamental research. For the fundamental research, you try to answer so many whys of the proof and mechanism. But in applied sciences, actually you have to stick to those keywords. That means your kits must be accurate, specific, sensitive, simple, rapid, cost-effective, user-friendly. And there is a need for the kit, and also it can solve a problem, and is very effective and reproducible. So you just need to memorize those keywords if you are interested to do translational research in future. So this is a 10-step translational research model that I have been used. It's actually my own model. It's just an example. So first of all, you can identify the key issue. That means you identify the problem, then you determine the output. Then you have to come up with the idea how to conquer, how to solve the problem. So from there, actually, you have to search for current development on the idea. You can search through internet or whatever, see whether there are similar inventions or applications or approach in the market. So no point inventing the wheel, just reinventing the wheel. Actually, no point. You have to create a new platform. So determine invention, platform, and design is very important. So in this case, actually, you have to consider a lot of factors. For example, the geographical areas, whether you need the equipment for such applications or all sort of things. Actually, you have to consider. So you have to certainly, especially the budget cut now, without any financial support, actually we cannot move. So you have to search for development financial support. This is very important. Unless you are so rich, you use your own pocket money to do the research. Okay, so we proceed. If everything's are in place, then you can proceed to develop the prototype, and further evaluations need to be carried out for the prototype. For the evaluations, actually can be carried out through 3 phases. That means lab phase, that means evaluate by your own scientists or your team, then further on by the field evaluations. Then after that, you have to obtain the certifications from authorized government agency. Okay, so that's the up to number 6. Actually, you have your prototype ready. That means something something is applicable to the society. Now it's up to you whether you want to patent your invention or you just want to keep it as your trade secret. Let's say the invention is so novel and nobody can duplicate the invention, so you can always choose the other choice. Okay, so after you have all this, you have to promote your invention and search for investor. This is very important because without the partnership and also the business model, your invention, it just leave it on the desk and it's just an invention, but you cannot really market it, you cannot push out the kit. So you have to determine, you have to sit down with your partner, draw out the business plan. Then after that, actually, finally you have your product. Then a lot of things you need to do, and a lot of hurdle you face after that. Okay, so let me bring you back to 2008. That was the moment we started to do translational research on malaria. And in 2011, actually, we successfully released Plasmonex, which is a malaria detection kit, into the market. So for those who are not familiar with malaria, this is actually a brief introduction on malaria. Malaria is a mosquito-borne parasite caused by Plasmodium. Plasmodium is the parasite that infects cells and transmitted through female Anopheles mosquito. So there are more than 200 species of Plasmodium are found in mammals, reptiles, and birds, but only 5 human malarias are recognized so far. That's P. vivax, falciparum, malaria overlay, and knowledge. Luckily, it's not so complicated. It's just 5. Okay, so this is the global impact of malaria. Malaria is one of the most severe public health problems worldwide and affect lots of children and pregnant women, especially in Africa region. So globally, malaria is the 5th cause of death from infectious diseases after respiratory infections, HIV, diarrhea diseases, and tuberculosis. So in Africa, malaria is the 2nd leading cause of death from infectious disease after HIV. So this map actually shows you the endemic and hyperendemic areas of the malaria. So the African region accounted for most global cases of malaria is around 88%, followed by Southeast Asia region 10% and Eastern Mediterranean region 2%. Okay, so this is the latest WHO report. Based on the report in 2014, there were approximately more than 400,000 malaria deaths worldwide. Most of the deaths happen in the Africa region, as you can see, because it's the hyper-endemic area, followed by Southeast Asia region, and the Eastern Mediterranean region is only 2%. Okay, so this is a reported malaria cases in Malaysia. As you can see along the year, the reported cases are going down from 7,000 plus to 3,000 plus. Because this is due to the malaria control management, and there's a slight increase in the reported cases for last year, 2014. Is close to 4,000 cases. All these are reported malaria cases. There are many others unreported malaria cases. Okay, in terms of financial contributions of malaria control, you can see funding for malaria control is around USD 300 million per year. This is actually the latest information obtained from WHO report. So when you look at the morphology of Plasmodium, which is the parasites for malaria, actually it's very complicated. But some of the audience here actually managed to identify, but not me. So to me, everything looks the same from one species to another. So especially they have different stages, and when you look at the blood film under the microscope, actually you can't really tell which is which species really infect the particular patient. So therefore, we try to develop something is simple. But first of all, you look at the species distribution and disease severity. In this slide, you can see P. vivax actually encountered for more than 80% of the global malaria cases, but the severity is mild. The most important species for Plasmodium is actually the falciparum and the knowledge side, because immediate treatment supposedly need to be given to the patients if the patients are infected with these 2 Plasmodium species, because treatment is delayed beyond 24 hours, the patients may die. Okay, So early detections and prompt treatments are needed, and misdiagnosis is actually not allowed, especially when it comes to these two Plasmodium species. Okay, because of that, actually, we came to a conclusion, and we have the idea in 2008 to develop a simple and accurate detections approach for malaria. Okay, so we started diagnostic methods comparison. We just see what other methods have been used in the market, and we also extracted out those data from the published paper. We found that actually each individual method itself has advantages and disadvantages. For example, microscopy actually is cheap enough for you to carry out the detections or diagnosis, but the skill level is high. That means you need to have a trained or experienced MLT to carry out the detection based on their naked eyes, based on their experience, based on their definitions or whatever. So for rapid tests, it's something to do because the specificity is either you can use that to detect Plasmodium falciparum or non-Plasmodium falciparum. So the species to be detected is some sort like limited. So it doesn't cover 5 species of the Plasmodium. So for nested PCR, certainly is good, but it takes a long time to obtain the result because you have to carry out the PCR twice, and the time for result is roughly 24 hours. Let's say you are dealing with many samples, this one is going to be very laborious, and the skill level is considered high as well. So therefore, we try to come to a conclusion, or we try to develop a single step multiplex PCR with our intention to reduce the time for result, to reduce the cost, to at the same time maintain high sensitivity and specificity for the kit. So the multiplex PCR is targeting Plasmodium species 18S SSU RNA gene. So the whole process is quite simple. That means you get the DNA from the patients, from the blood samples, then you just carry out the multiplex PCR screening, then followed by the result analysis. So this is actually the gel view for the result. So as you can see, you no need to recognize the Plasmodium just based on the morphology of the Plasmodium, but you just need to recognize the species based on the band size. Okay, so we did carry out sensitivity tests as well. This is to determine the detection limit. We also carry out the specificity test by using DNA from other organisms, parasites, and also bacterial genome. Okay, this is to show you this kit actually is able to detect the mixed infection. This one will complicate the diagnostic for Plasmodium if you are using microscopy approach, because it's some sort of like complicated, confuse you. So the MLT become not so sure whether it's 1 species or 2 species or 3 species of the Plasmodium are infecting the particular individual. So end of the day, actually, we come to the final product, and this is the feature of the final product. It's able to detect 5 species of Plasmodium, the human Plasmodium, and the result is actually within 3 hours, and the skill level is moderate because as long as people can run PCR and followed by gel electrophoresis, actually it should be fine. And most of the postgraduate nowadays actually is able to do that. And in terms of sensitivity, actually it's 5 parasites per microliter of blood. That means that's a detection limit. You need to have 5 parasites in 1 drop of blood, then you can use this kit for detection. Okay, this would like to share with you my family members. Today only my wife is with me now. My parents is in Johor. It's not so convenient. Okay, but their hearts are always with me. They provide the best to me all this while. But one thing I never listen to them is they don't want me to become a teacher. It's too bad because the family members, all my family members are teachers, ranging from primary school to even my aunties, my uncles are teachers. So too bad I become a teacher at last. Okay, so that's my three cheeky kids. In fact, actually, all of them some sort like make my life so balanced, and they have lightened up my life so far. And my wife purposely take leave today to accompany me, although she didn't sleep at night last night because of the work. Okay, so I love all of them all this while, and they really give me the full support all this while. Okay, awards. Okay, I just put on hold first. We'll be presenting to you later because I want to share with you something else which is more light and not so taxing to you. This is how did I awarded myself after each exhibitions in overseas. It's just a selective slides. Okay, so this is in Germany. This is a setup in Germany. And this is the church. In fact, the buildings are looks alike when you stay there for more than 2 weeks. This should be a church. Okay, this is market in Nuremberg. So after that, actually, I take my own leave to travel from Nuremberg to Berlin. This is fantastic. This is actually called Berliner Dome. Okay, it's the church in Berlin. And this is the Berlin Wall. Okay, and that's the actually Victoria column. Okay, in fact, I buy the Berlin the stone from the Berlin Wall, but people say I get cheated with a few I think more than €10 a piece of stone. They say the stone actually is not from the. I think my wife said the stone is not from the Berlin Wall; it's from the roadside. But still keeping somewhere in my house. Okay, it's very costly. Okay, in Hong Kong. Okay, this is a setup in Hong Kong. It's quite advanced over there because there are many investors from Shenzhen to China to exhibit their product as well. So these are the Malaysian exhibitors from various institutions. So we go together. We are so happy to see each other there. But everybody just split up after that to go to sightseeing for their own destination. Okay, so after that, actually, I managed to go to Ocean Park. Managed to take a picture with Gloria. I think that's her name. I guess my kid told me that. Okay. So at the same time, this is official visit to Dolphin University. This is fun. They set up an area to train the dolphins for performance. So this is the Dolphin University in the Ocean Park. So one day, let's say you have the opportunity to go over there, you can always visit. So it's quite boring to go to. I visited Pittsburgh University and other university, but this is actually quite fun. Dolphin University. Okay, this is Rantau Island. So in Korea, this is a setup in Korea. Then this is the museum in Korea. This is in Seoul. Okay, so this is one of the palace in Korea. So the organizer are some sort like quite nice because they want to promote their culture. They some sort like organize a half-day trip for us, bring us to all those areas, hotspots for us to pay a visit. This is actually the Seoul City from the peak. And this is actually very beautiful. It's Winter Sonata. And one day I wish I can go with my wife there. I can't show those pictures here because I was with someone else between the trees. Okay, but I think she knows that. She purposely dig out those clips of photos from my computer. Okay, so this is in US. This is a simple setup. It's just using the cloth. Okay, so you just stick on your poster on the cloth. Then this is the setup. And this is a team of many kids. Okay, Universal Studio in LA. Okay, so we managed to spend half a day there. Okay, try to enjoy all the rides. Then this is Hollywood. Okay, and this is the street in Hollywood, the scene in Hollywood, and this is the Walk of Fame. But I can't find my name there. Okay, I can find even Snow White is there, and but I can't find my name there. Okay, it's quite sad. So since we are in Hollywood, I would like to share with you a video, a show.
Raise the flash, gotta know my age. Know my age. Know my age. My ooh, my oh, my oh, my age. My ooh, my oh.
With that, thank you for your attention.
Distinguished guests, I would like to call upon Professor Dr. Adiba Kamarul Zaman to conclude the lecture which has been delivered by Professor Dr. Chua Kek Heng.
Thank you very much, Prof. Chua. And in his usual manner, he was very humble and almost apologetic for all his achievements and awards he's won. You may not have your name next to Tom Cruise, but I think when builds his own Walk of Fame and Wall of Fame, I'm sure you will earn a place very high up on the list. And Mrs. Chua, how big is the room that you had to build to place all his awards? I think that alone, you probably have to build a special room just to display all the awards that you've gained. Nonetheless, I'm sure you will all agree with me that it is not just the awards that you have received that indicates the level of achievement that you have acquired as a scientist and a researcher, but truly, I think, as I said earlier, what the difference between your research and many of other researchers, many of our colleagues, is that your research, the translation of your research has meant that the diagnosis of malaria, something I'm familiar with, has been simplified and can really, truly mean the difference between life and death, particularly those with Knowlesiae and falciparum malaria. So that's just one example of the kind of bench work that is meaningful and impactful. And I think as a son of University Malaya, I'm sure others at are truly proud of you, not just in the awards that you've received, but also I'm sure it came across very clearly in the presentation this morning, not just his humility, but also his passion and care and concern for the issues and the diseases that he's studying, and behind it all is the real desire to make a difference. So thank you very much, Prof. Chua.
Thank you, Professor Dr. Adiba, for chairing this lecture. I would now like to take this opportunity to thank the management of the Faculty of Medicine, International Corporate Relations Office staff of as well as all the guests present here this morning. I would now like to also invite Professor Dr. Chua Kek Heng's students to present him their gifts of appreciation. With this, we have reached the end of the lecture. On behalf of the University of Malaya, I would like to apologize if there has been any weakness from our side in the organization of this lecture. Refreshments will be served upstairs at Synapse and in the foyer of Dewan Kuliah 1. Ladies and gentlemen, once again, thank you and have a pleasant afternoon.